All right, ladies and gentlemen, welcome back. We are going to continue talking about viral replication and talk a little bit more in depth about the replicative cycles of animal viruses. So, when we look at viruses that can infect animals, there really are two key variables that end up being utilized to classify those viruses and then be able to understand the way that they typically infect. And the first thing to just figure out really is what type of genetic information do they have, and then in what form does that genetic information come in. So we want to talk, are they DNA versus RNA viruses? And then we look at, are they single stranded or double stranded? And that's really going to drive the way in which they need to utilize their host cell's machinery once they get in and actually infect that cell, the way that they use that genetic information then to be able to continue building more of themselves. So if you take a look at the table there, just gives you some examples of certain viruses that you may be familiar with that fall into one of those four categories. So we can have DNA that's typical double-stranded, like most students would think of, things like pox viruses, so smallpox, chickenpox. DNA can also be single-stranded. All right. RNA can be double-stranded, which is something that most students don't think of. Retroviruses that are pretty commonly connected to things like diarrhea, those are double-stranded RNA viruses. And then you can have that traditional RNA single-stranded that kids think about. So things like rhinovirus, which causes most common colds, so on and so forth. The other thing is to recognize whether or not those animal viruses contain envelopes, because that will make a difference on what they end up doing to escape the current host cell that they're infecting. If they don't contain an envelope, they'll typically just continue to build and build and build and eventually just burst out of their host cell. But a lot of viruses that infect animals are contained within this membranous envelope. So you take their genetic information here in the center, you get some sort of capsid protein coat, and then you have an envelope exterior to that. And then most of the time, those envelopes contain other carbohydrates and other proteins that come off of that essentially phospholipid membrane that's surrounding their protein coat. And those viral glycoproteins, glyco being carb, protein being a protein, so it's a combination of the two, on that envelope will then bind specifically to receptors on the surface of other host cells and allow them to gain entry. So this is a really clever way that these animal viruses have evolved because they'll use their host's natural receptors that are necessary for that host cell to communicate with other cells within that organism's tissue and coordinate function. Um, and then they'll also take them with them when they leave, um, so that they are much more easily able to infect their next host because they're molecules that the host already contains and uses for other purposes. Um, So what they'll do is they'll utilize those glycoproteins attached to receptors on their host cell's membrane that will induce what we call receptor-mediated endocytosis. So endocytosis, remember, is the surrounding of the cell membrane of a cell and using it to take in large solid particles. So they do this to sort of eat, if you will, all the time. So those viruses have taken advantage of that mediated endocytotic process to gain entry. Once they gain entry, they'll go through and do their replication of their genetic material, building of their capsid proteins, taking over the host as a whole, and then they will also, in that process, allow the host cell to continue making those glycoprotein receptors that get moved from the ER to the Golgi to vesicles that eventually merge with the cell membrane, so that when that fully assembled interior component of the virus is ready to go, it will go through exocytosis and take some of that host cell membrane along with it to become its new envelope. when we look at the variety of viruses that exist, the broadest variety really is found in RNA genomes, especially in viruses that infect animals, which might come as kind of a surprise since most of our genetic material is being driven by DNA. One of the kind of interesting RNA classes are these retroviruses, and the one you're probably the most familiar with is HIV or AIDS. Um, so retroviruses, not only do they contain their genetic material, they are an enveloped virus, so they have that glycoprotein lipid membrane, they have the capsids on the inside, but they also carry the enzyme called reverse transcriptase. And it's if you break that word down, that enzyme name down, well, the ASE should be recognizable to you as an enzyme, and it's reversing transcription. So if you remember the flow of genetic information in cells, it's typically DNA to RNA to protein. So translation goes from the RNA to the protein, but in this case, we're reversing transcription. Transcription typically goes DNA to messenger RNA, and instead we're going to reverse that transcription process and go from RNA to DNA. And that's exactly what's happening here. So that infection pathway releases not only the genetic material inside the host, but also that reverse transcriptase enzyme that will then take that RNA to DNA, and that DNA actually gets incorporated into the host cell's DNA and then utilized to go through traditional transcription and translation to then build new viruses that then bud off. We'll talk a little bit more later on about HIV specifically, so we'll come back to this pathway. But for now, just recognize that viruses can contain not only their genetic information, their protein coat, and sometimes that envelope, but some of them will bring some unique enzymes or other molecules to help them along their infection pathway as well. And retroviruses are an interesting one because they'll actually go the opposite direction in order to incorporate themselves into their host. Proviruses, while we're on the topic of getting that genetic information into its host's genetic information, sort of similar to that idea of a lysogenic cycle in a bacteriophage. Viral DNA that is integrated into a host genome that is not a bacteria, we talk about that as a provirus. So when we look at something like HIV that goes and infects, uses that reverse transcriptase to make that copy of DNA of its own genetic information. Once it gets incorporated into the host cell's DNA, we talk about it as a provirus. And then it is that proviral DNA that is being transcribed and then translated to make more HIV viruses or any virus that would work in the same pathway. So that RNA that it's bringing in with it works kind of in two ways. It will work both for, I should say, the messenger RNA that's getting produced from that provirus will work in two ways. It will just become more RNA that will end up getting packaged into those new viruses, but we can also then use it as instructions to build those capsid proteins as well.
AIDS is caused by the human immunodeficiency virus, HIV. This is an enveloped virus. The envelope is derived from the host cell membrane with viral glycoproteins such as GP120 and GP41 inserted into the membrane as the virus leaves the cell. The core is composed of P24 capsid protein, and the P17 matrix protein helps maintain viral structure. Inside the core are two identical copies of single-stranded RNA viral genome and three enzymes, reverse transcriptase, RT, protease, PR, and integrase, IN. To establish infection, HIV must first attach to its host cell. Attachment occurs by interaction between GP120 on the surface of the virus and the CD4 antigen receptor on the surface of the host cell. In addition to the CD4 receptor, there must also be a co-receptor on the host cell. The co-receptor differs for different host cell types. In T lymphocytes, the co-receptor is called CXCR4. whereas on macrophages, the co-receptor is called CCR5. After attachment, the viral envelope and host cell membrane fuse, resulting in entry of the virus into the cell. Once the RNA is released into the cytoplasm of the host cell, reverse transcriptase makes a DNA copy of the viral RNA genome. As the DNA is being formed, reverse transcriptase degrades the RNA strand. A complementary DNA strand is then added by the reverse transcriptase, and the ends of the resulting double-stranded DNA segment are joined non-covalently. Treatment with nucleoside analogs or reverse transcriptase inhibitors interferes with these steps. The resulting circular DNA is then moved to the nucleus and inserted into the host cell chromosome by the viral integrase, IN, enzyme. The integrated viral DNA is now referred to as proviral DNA. Following integration, the proviral DNA may remain dormant or, with host cell activation, RNA may be synthesized from the DNA, yielding messenger RNA and viral genome RNA. Viral messenger RNA is translated, yielding viral enzymes and structural proteins. Some of the functional proteins are formed by cleavage of a long polyprotein by the enzyme protease. Protease inhibitors interfere with this step. GP41 and GP120 are inserted into the host cell membrane, and the structural proteins surround the viral RNA to form the core. Finally, the virion is released by budding.
So I've sort of mentioned interesting things that have occurred, generally speaking, in viral evolution. But there's sort of an interesting breed of thing. Viruses don't fit that traditional definition of living organisms by fitting all of those characteristics of life you probably were taught as underclassmen: the metabolism, the response to stimuli, the reproduction, the evolution, so on and so forth, because they are required to be within a living cell and using that cell as a way to gain material to make more of themselves. So they probably evolve in a very interesting way in and of themselves, given the fact that they have to be within that host and they're utilizing that host very effectively as a way to replicate. So when we look at sort of mutations and things that occur in viruses over time, as part of that evolutionary pathway, candidates for the sources of those viral genomes can come from a whole host of places, and more often than not, they've probably come from bits of organisms that they have infected as a species, if you will, a class of virus. They're not really species if we don't consider them living things, but coming from bacterial plasmids, from bacteria, circular DNA, yeasts, transposons, which we'll mention a little later this year, and these are just small little components of DNA in a lot of organisms that are capable of moving. So lots of places for viral genetic evolution to come from. So it's kind of interesting if you start to dive into their genetics, because it really is kind of a hodgepodge of places we think that stuff has come from.